So animal breeding is an example of biotechnology that has existed for many centuries. Vaccines, we talked a little bit about the origin of vaccination, and how a specific disease called smallpox really was the beginning of our quest to begin to use vaccines. But vaccines, especially in the 20th century, were important as the world wars really introduced these diseases to on a global scale that had not been achieved before. So um, vaccines really erupted as a result of the fact that many parts of the world were now experiencing diseases that they hadn't experienced before, like smallpox, yet again. And so we developed vaccines in order to prevent pandemics from breaking out. Cloning is not something that happens very often. And of course, this could present an ethical issue. And we're going to talk about the ethics of biotechnology later this week. But cloning is possible. Antibiotics are an example of biotechnology. GMOs are genetically modified organisms, which are, some people consider them to be less healthy than food that is grown more organically. Pest-resistant crops, which of course mean crops that are maybe even poisonous to pests, so they just over time evolve to stop eating them as much. Stem cell therapy, gene therapy, designer babies. You may be wondering what that is, but there have been examples in which people have been able to, well, at this point we have the scientific ability to literally edit a baby's genome so that that baby will have a specific eye color or a specific hair texture. Of course, that is an ethical issue. That presents an ethical issue as well. And then there are many others that we won't specifically. Go ahead.
Is that somebody's brain?
No, that's probably some type of meat that has been genetically modified or grown in a petri dish. So this is something that is coming up, as well as many people are recognizing that agriculture, especially the amount of land and water and food that is dedicated to agriculture, specifically to cows, is just not good for the planet. And so the amount of beef that we're consuming is actually pretty unhealthy, not really just for us, but also for the planet. And so these companies now are developing ways to grow meat in a lab. And it's not really the exact same thing as, like, it's actually not at all like a black bean burger or anything like that. It's actually lab grown meat. So this is something, there's actually a good show on Netflix that talks about this. I can't think of what it's called, but if that interests you, then you can do some research and find it. But yeah, that's becoming a thing now. Okay. But today we're going to talk about DNA fingerprinting. We'll talk about some of those other things tomorrow and on Thursdays as we get into the ethical issues of biotech. But DNA fingerprinting is the process of determining an individual's or a species' DNA. Now, DNA fingerprinting allows us to sometimes establish a link between biological evidence and a potential suspect in a criminal investigation, but it's not perfect. It is not a foolproof scientific method. And there are many conversations about whether or not it should even be used in courts. I think when we're younger, we're told that everybody has a unique fingerprint. That's largely true, but oftentimes when you touch something, you don't leave a complete fingerprint. There might be, you're putting more force on a certain part of your finger than another part. And so often the fingerprints that can be collected from a crime scene are partial fingerprints, and a partial fingerprint is, can be shared by multiple individuals. So again, there's some ethical issues here as well. But DNA fingerprinting is still pretty useful. And the methods that we use for DNA fingerprinting are also used to determine paternity tests. They're also used to determine the similarity of species. So this is good technology. Okay, moving on in 3, 2, 1. So we can use DNA fingerprinting to compare DNA data. So what you're seeing here are fragments of DNA. This is a technology or a methodology that is very, very common. And those of you who are interested in studying science in college, you would actually go into the lab and do this as a part of your freshman year biology course. So this is pretty cheap. It's a process called gel electrophoresis. Let's see if we talk. Yep, we'll talk more about it on the next slide. But it allows us to separate the DNA so that we get fragments. And then we can compare, let's say we've collected data from the crime scene and we've got a number of suspects. We can compare the data that we've collected to the known DNA of the suspects to determine a match based on the number of fragments that they have in common. So the more fragments in common, the more likely the DNA is from the same individual. So how do we get this DNA fingerprint? How can we really read it? So the answer is gel electrophoresis. This is something you should write down. This is maybe a difficult word to pronounce, electrophoresis. But it's a process in which the DNA fragments are separated by size. So we've got this sugar called agarose. Remember, sugars typically end in OSE. Enzymes end in ASE, but sugars end in OSE. So I mix this sugar with a buffer, and a buffer is something that can neutralize the acidity of a compound. I boil that mixture. I then pour it into a mold here, and I let it cool down. It'll become a gel, so it's not quite solid, but it's not quite liquid either. And the gel is separated into different wells. And so I can then load my DNA into each of these wells. Let's say I've got 3 sources of DNA. I put one source in this well, another source in this well, and a third source in that well. Gel electrophoresis is, you see this prefix electro. That's because we use a source of charge. We use a source of electrical charge to actually pull the fragments, the DNA fragments, through this gel. So I literally flip a switch, it turns on this electrical charge. One end of the gel is going to be positively charged, the other end is going to be negatively charged. And over time, I can see that the smaller fragments will move further through the gel. The larger fragments are just too big to fit, so they kind of stay at the top. So I get this nice separation as I move through the gel, where all the smaller fragments are at the bottom and the larger fragments are at the top. And then I can compare them and say, well, I see that this fragment that's at the top of well number 1 is also at the top of well number 2. And this fragment in the middle of well number 1 is also in the middle of well number 2. And then if I can determine that there are enough similarities, then we can make a conclusion that the DNA have come from the same organism. So electrophoresis is a very simple process. It's cheap to do. It doesn't take much time at all, but it ended up being very important for our ability to identify organisms and species. So nothing to really write down here. You all know this already. The sequence of the nitrogen bases determines an organism's DNA fingerprint. So DNA is unique because each of us has very slightly different sequences, right? So Ayana might have GTTC as we see here, and I might have GTTA, and Alicia might have GTTT, and Malachi might have ATTT. So there are small differences that you will see in very specific places in our genomes that make all of the difference in terms of who we are. But of course, this is totally dependent on the sequence of the nitrogen bases. So here's what it might look like. Here's what a gel might look like. The M's represent the known DNA, and then we've got 1, 2, 3, 4, 5. So 5 other sources of DNA. And then we can literally compare. So all 5 sources share that same top fragment. It looks like both source 1 and 2 share this 4th fragment right here. And so on and so forth. So on and so forth. So you can compare them to identify the DNA. So we can use this process of gel electrophoresis to identify suspects in potential crime. We can use it to identify paternity, so we can find out who the father of a child is. And we can also use it to catalog endangered species. So let's take a look at this. If I've got crime DNA data right here on the left side, and I've got suspects right here, which one of the suspects is most similar to the crime DNA?
Suspect number 2.
Good. Suspect number 2 is most similar to the crime DNA. We see they've got this top fragment in common. They also have this fragment in common, the second fragment. They have the third one in common, so do suspects 3 and 4. And they have this last fragment in common, just like suspect 4 does. So suspect 2 has all 4 fragments in common, which means that it's more likely that suspect 2 is the source of this DNA. Can we determine who the similarity is over here on the right side?
Can you repeat that?
Yeah, which suspect in this DNA electrophoresis is most likely the source of this crime scene DNA?
Number 2.
Good, number 2, yeah. So we can literally just see, and this is what it would actually look like, you know, if we were to take an X-ray of the final product of the final gel electrophoresis, but we can see that the fragments perfectly match up between the crime scene DNA and the suspect DNA, suspect number 2's DNA. All right. So another application is paternity testing. So can we determine which person, which man over here is the father of this child? Keep in mind that obviously the child is getting half of its DNA from its mother as well. So when we're trying to identify who the father is, the father does not need to have 100% of his DNA matching the child, because obviously the child is getting some DNA from their mother as well. But can we determine based on the fragments here who might be the father?
I'm gonna say dad number 1.
I say dad number 2.
Any other thoughts? Any other?
Never mind. I don't know which one to say.
Okay, so the way that you do this is a bit trickier. Just a bit trickier. Because this one, yeah, it's just a little more complicated. So what you'll want to do is count, literally just go fragment by fragment, and then add and figure out how many fragments are in common for each dad, for each potential father. So this top fragment here, how many of the men have that fragment? Can you all tell? I wish there was a way for me to do like a ruler.
Is it dad 2 and dad 3?
Yeah, dad 2 and dad 3 both have that fragment. So that means that if we can just kind of keep a running tally in our heads, dad 2 has 1 fragment now, dad 3 has 1 fragment. What about this second fragment? Any thoughts about this second fragment here? Maybe you all are stuck because it doesn't look like any of the potential fathers have that fragment. But if we
Yeah.
look to the left, the mother has that fragment. So that makes sense. The mother contributed this DNA to the child. Okay, so that's why paternity testing is a bit more complicated, because you do get the child obviously has 50% of their DNA coming from the mother. Let's jump down to this third fragment here. Which of the fathers, or the potential fathers, has this fragment? It's hard for me to move in a straight line with this. Okay. Well, it looks to me as though dad 1 is the only man who has this fragment. Dad 2 has a fragment that's close, but it's not in the exact same location. So now if we're keeping a running tally, each of them has 1 in common so far. So let's look at the last 3. I can see that this fragment here looks to be shared by father 1 and father 3. So now they have 2 each, and dad 2 only has 1. This fragment looks to be shared by only dad 2. So now all of them have 2 fragments in common. So it really comes down to this last fragment, which looks to be shared by dad 1. Oh, actually, did I forget? So 1, 2, 3 for dad 3. 1, 2 for dad 1. 1, 2 for dad 2. So it is dad 3. Sorry about that. Dad 3 has the top fragment, this third fragment, and this fifth fragment in common with the child. So dad 3 is the father of the child. This is much easier when we can actually use rulers and draw lines between them. So this is going to be a little challenging to do electronically.
Oh, sorry, Mr. Red. So is this what they do? You know, on the shows where they have like the parents and they have to find out who the parents are and stuff?
Yes, yes. Yeah, this is the exact same process. It's obviously more complicated. You've got many more fragments to compare, but this is how they do it. So we can see that these lines here, this is what we would be doing if we were in class, literally just drawing lines and counting how many of the lines match up. So dad 3 ends up matching with 3 of those fragments. So he's got the most, he must be the father. We won't say too much about how the mother didn't know between 3 different people who the father might be, but to each their own, you know? Okay. All right. Okay, do I want to have you all write this down? Let's see. Okay, so go ahead and write down the steps of creating a DNA fingerprint. So obviously the first step is to extract the DNA. So you need some source of DNA, whether it be hair, saliva, blood. You need to be able to get DNA, which we know, of course, is housed in the nuclei of cells.
Hey, Mr. Rudd.
Yes.
My canvas still isn't working. So what do I do?
What is it? What's it telling you when you are you able to click on the canvas?
It'll be open for like 30 seconds and then it'll close and say I'm denied.
Okay. So I will send an email to our tech director when we're done with this lecture, and then I'll copy you on that email, and I'll just ask if there's anything that he can do. It's likely because you are a new student, but it should be cleared up by now. So I'll send that email in the next 10 minutes or so.
All right.
So once you've extracted the DNA, you need to cut it into those fragments that we were just talking about. The way we do this is with use of an enzyme. It's called a restriction enzyme. These enzymes are pretty cool. They are like scissors, but they are scissors that only work in specific places in the DNA. So they'll recognize a nucleotide pattern and cut the DNA at that specific location. Then we use that process that we just talked about, gel electrophoresis, to separate the DNA fragments. So after they've been cut, now we need to separate them, and we do this using an electrical current. The larger the fragment is, the less able it is to move through the gel, so it will stay at the top. But the smaller fragments can travel further through the gel, so they end up closer to the bottom. And then lastly, you just compare the results. Okay. Moving on in 3, 2, 1. So again, this is what it looks like. You've got a power supply, which you literally just switch on. It's sending a negative electrical charge to the bottom of the gel and a positive electrical charge to the top of the gel. There are going to be a number of wells in the gel that you can load DNA into. So I'll just put all of these fragments of DNA into this gel, and when I turn on the charge, the fragments will be pulled through the gel at different distances. Then I can use an X-ray to take a picture of the DNA and determine and compare the fragments and how they've traveled through the gel. So this entire process takes less than an hour. Again, if you are interested in studying biology, or if you're interested in studying any science or going to medical school, you'll have to take a biology class, and you'll do something like this in the lab component of the class. So you load in the DNA, you turn on the charge, the DNA moves through. Your longer, heavier fragments don't move very far. Your shorter fragments are able to move much further. And then you can kind of figure out, you can do whatever comparison you're trying to do at the time. These are called pipettes. They allow you to move very small amounts of liquids. So something as small as even 5.
That is so.
Yeah. So your restriction enzymes, again, they're like really specific scissors. They figure out the pattern in the DNA, and then they go in and they cut at that specific place. So this enzyme, an example here is a restriction enzyme that cuts after the pattern TAAT. So anytime it sees TAAT, it's going to go in and it's going to cut right after that. And this allows you to have fragments of DNA. So now this would be its own fragment. This is its own fragment. This is its own fragment. This is its own fragment, and so on and so forth. Okay. So if you're interested in law school, or if you're interested in practicing law in any way, then you'll probably hear more about the Innocence Project. You'll hear more about the legal admissibility of DNA fingerprinting, which means if something is admissible in court, it means that you can use it in some type of testimony, or you can present that evidence to the jury or to the judge. So prior to 1984, DNA fingerprinting was not admissible evidence. You could not use that as something to convict an accused person. That was largely because the science was just not yet proven. And then over the last 30 years or so, the science improved. But now the science has improved to the point where we know that DNA fingerprinting is imperfect. Okay, so where DNA fingerprinting has been able to exonerate or prove the innocence of some people, we also know that DNA fingerprinting is imperfect, and it can be used poorly, and it can be used to actually wrongfully convict people who are not actually guilty of crimes. So we're at this kind of weird point right now where the science has improved, and it improved originally, which made it easier for us to use the science and to convict people. But now we know that there have been mistakes along the way. But this Innocence Project is a nonprofit organization that works to free wrongfully convicted people using DNA evidence. So prior to 1984, DNA evidence was not really used in court. We relied heavily on what's called circumstantial evidence. So people who didn't have alibi, or if you could prove, or you thought you could prove the motive of someone, or if they matched the description of a witness's account of the suspect. These were the types of evidence that were considered in court in terms of convicting or judging a person's guilt. DNA evidence is obviously a bit stronger than that, because we know that people's memories change and people have specific biases, but the science does not have the bias, right? So the DNA fingerprinting was a big development, and the Innocence Project relies on that.
You still have that letter? Can I see it? This is it. You still have this. Is like, wow, this is, wow. Mr. Sheck, I'm confident that whenever the DNA test is done, it will show my innocence and this miscarriage of justice.
You are the best of the best when it comes to DNA evidence. This is pretty much known throughout the country, and I desperately need your help.
Greetings, I'm James Harden Jr. I'm writing you this letter in sincere hope that you could be able to help me and my brother, Jonathan Barr, regain our freedom.
My name is Jonathan Barr, and I was wrongfully convicted of a murder and a rape of my friend Katrisa Matthews, and I spent almost 20 years in prison for that crime.
I spent 15 years inside of prison.
I spent 11 years and 3 months in prison.
20 years, 9 months, 5 days in prison for a crime I didn't commit.
The system failed us, tremendously. It failed us.
What happened to this female is wrong, and the real rapist has confessed to attacking her alone. They allowed the victim to be shown mugshots. All the mugshots was in black and white with the exception of my ID. She basically focused on my ID picture, and when she saw me in the lineup, within 5 minutes after being shown mugshots, she identified me as her attacker.
It was a bite mark on the victim, and the odontologist said that my teeth was the one that made this bite mark.
There was DNA evidence that excluded us from being, the donors of the semen that was found inside of Katrisa. But I guess because they had these ironclad statements from co-defendants in our case, they still proceeded. We were prosecuted and found guilty.
I gave up in prison several times. I don't know how many appeals I filed.
I had just got denied parole for the 5th time, and one of the counselors that was working there, he pulled me. He wasn't my counselor. He just pulled me to the side. He said, "Look," he said, "Marvin, there's an organization out there that may be able to help you."
I had a lot of support from the Innocence Project. They were very, very helpful. That day I heard about the DNA testing and I was excluded, I was overwhelmed with joy.
It was a great relief to be able to walk out of there a free man after 14 years. I'm able to spend time with my grandkids. I can hold them in my arms. I can take them to church, take them out to eat.
My mom was suffering from dementia and congestive heart failure. Despite of what her body was saying, her spirit was like, I can't die until my son gets out of prison. And she was able to see me completely exonerated on TV. And right after that, she died. She died at peace.
I wish my parents would have lived longer to see this, but I know in spirit they're here and they will continue to be here for as long as I am here and as long as my daughter is here. And if I am to have any more children, they will live through them. You know, but I'm thankful. I'm thankful to be home.
I can remember my first day coming home from prison. I sat up and watched the sun come up that morning. You know, and you see the sunlight every morning rise in prison, but to actually feel it, you know, as a free man outside of the fences, it was a totally different experience. You know, the whole time I'm watching it coming up, I'm saying, I'm free. You know, I'm truly free. And without this organization, a lot of men and women who are free now would not have been freed. And I am one of those men.
Okay, so the assignment for the asynchronous portion of class today is available on Canvas. It is called the Innocence Project. Malachi, I am going to go ahead and send that email on your behalf, try to figure out if we can fix this issue. But your goal is to write a paragraph about one individual's case. So you're going to go to the link that is provided in the assignment. You're going to choose a case, and you're going to just type up a brief summary about how DNA evidence was able to prove their innocence. Okay, so it doesn't look like once you go into the assignment, it's not going to look like there is a place to actually type, but you have to click submit first, and then your text box will come up. He's not here anymore.
I have a question.
Yes, ma'am.
This has nothing to do with biology, but who do you talk to if, like, from one of my classes, we're required to have a website that we work on, but the school blocked it. So how do I, like, reach out to them to tell them, like, ask them if they can unblock it, or the teachers need to do that?
Yeah, your teacher probably. I would start with reaching out to the teacher who
Okay,
assigned that. Yeah,
thank you.
you are welcome. Okay, folks, it is 11:35. So at this point, you all can continue working on your asynchronous assignment. It shouldn't take you beyond this lunch period, but I hope you have a good rest of your Tuesday. We will wrap up Unit 6 in just a couple of days. I appreciate you, Castro, for reminding me to record, and I hope you have a good rest of your day.
Adios.
Adios.